Thank you again, both for the invitation. It's actually, I'm really happy that I can have this joint session with Hannah. We discussed beforehand, and we decided that I will give an overview of what we know so far from the published literature about genotype and phenotype associations in PCD, and Hannah will go in with her expertise at the second part of the session, where she will explain more the pathophysiology of some of these mechanisms and associations. And before I start, I will not stay very long at this slide. And I just wanted to remind to everyone that we know that in PCD, mutations in numerous genes, code, proteins, that they are involved in different stages of the ciliary assembly, the structure and the function, and they have been grouped based on the different type of proteins that they are coding. And this is the basis of the whole discussion that we are having about possibility of associating genotypes with phenotypes. When we discuss about geno-pheno associations, it's very important to understand and clearly what each terms means, because in many published studies, this is not always straightforward and clear, especially because the difference between the functional phenotype, which is actually the ultrastructural phenotype, and the clinical phenotype associations, it's not always described. So for me, as coming from an epidemiological and clinical background, when I talk about the genotype is clear, it's the combination of alleles for a specific affected gene. But when I talk about phenotype, and this is where I will focus today, I'm referring to the clinical phenotype. In case of PCD, we also have the functional ultrastructural phenotype, which is the actual type of ultrastructural defect that is caused by the genetic mutation and that leads to the dysfunction of the cilia and the disease. And the clinical phenotype, what we will refer today, it's all the combination of different traits and manifestations, which includes not only symptoms, but also other expressions of disease, such as functional and structural disease seen by lung function, for example, or imaging in different organ systems. So throughout the presentation today, there will be reference to the ultrastructural phenotype, because that is something that plays a very important role in all the genotype-phenotype association studies in PCD. But when we refer to phenotype, we will mean the clinical one. So we know, and that was something that was shown first before we started discussing about clinical phenotype associations, that specific gene groups are linked to specific structural elements and specific ultrastructural defects. This is an overview of groupings. This is already has been updated. It is from a few years ago, of different genes where their mutations lead to outer DNA arm or outer and inner DNA arm defects, or defects on the central apparatus and radial spoke, or even to normal infrastructure via electron microscopy. The reason that I'm showing this is that because a lot of the studies that I will present, when they discuss genotype-phenotype associations, instead of referring to the specific mutated gene, to the specific genotype of the patient, they use the corresponding ultrastructural defects grouping. And that makes it easier and allows them to have larger number of patients to study this kind of associations. One of the most straightforward associations that have been studied in PCD is associations, genotype associations with laterality defects. And the reason that this is very straightforward is that we know that the nodal primary cilia do not have radial spokes and the central pair structure. For this reason, it's an easy thing to observe and understand that mutations in genes that they have been involved in the protein structure of the radial spoke or the structure of the central pair do not have an effect on the nodal cilia, and they do not lead to laterality defects. And that has been studied in several of these genes, and is something that we know and we expect. So now this might have been the only clear and straightforward thing that I will show you today, because going into respiratory defects and the respiratory clinical phenotype, the things are slightly more unclear. The first studies that start and still continue assessing genotype-phenotype associations are actually genetic studies that describe new genes, and usually they focus on a specific population of patients with a specific newly described mutation, study if this mutation exists also in the family, and then they describe with as much detail as possible the functional phenotype, the ultrastructural defect, and the clinical phenotype. So these are some examples of studies that have been published in the past 9 years, and some are a bit more recent with different genetic mutations where it has been shown that they can lead to laterality defects, for example, DNA H9, but they have overall a milder respiratory phenotype. But this comparison is not a direct comparison with patients in the study, but overall with what we know as the typical PCD phenotype from the literature and from clinical experience. Another genetic mutation that has been associated with a milder disease and also higher nasal NO values is the RSPH1 mutation, and that has been reported that patients with that had a lower prevalence of neonatal respiratory distress and the later onset of chronic cough and better lung function later on. While patients with RSPH9 mutations, and this is also quite a common example, they had a very wide phenotypic variability. So in that case, we cannot safely say that this gene is associated with some specific characteristics and clinical phenotype. Besides the genetic studies, what we try to do mostly in the literature is to really use large databases and large, usually cohort studies or registries, to describe using an increased number of patients where we can have enough patients with the different subgroups of genotypes or at least functional phenotypes to describe differences in clinical characteristics. This study from the North American Consortium, it was, as far as I know, the first larger cohort study that described the first baseline cross-sectional data comparing patients with DNA in arm defects with patients with central apparatus and microtubular disorganization defects. They did not find, that was in children, and they did not find any difference in the prevalence of clinical symptoms or in the nasal NO levels. However, they did find that patients that had central apparatus or microtubular disorganization defects, they had worse FEV1 and FEV25-75, so worse obstruction. So in this graph on the slide, the patients with square, either white or filled in, the ones with the colored squares and circles are the ones with the known genetic mutation. They are the ones that they have, the squares are the ones that they have the lower lung function in comparison with the circles that corresponds to patients with DNA arm defects. They also studied radiographic disease. And here the findings were a bit less clear, but still, especially in the age group from 5 to 11 years, they described that the patients with microtubular disorganization defects, they had more lobes affected with bronchiectasis and alveolar consolidation compared to patients with DNA arm defects. This was not the same throughout their whole population. However, what the authors highlight and what is very important to keep in mind for this kind of studies is that the numbers of the subgroups were very, very small in some of the subgroups. So, for example, the subgroups of older than 12 for microtubular disorganization was, I think, 6 patients. So sometimes this kind of comparison, even when we group the genes, is very difficult to do. And they also described that these patients had purer growth, or they had a lower weight and height in comparison with the patients with DNA and arm defects. The lung function is an outcome that has been studied very consistently in phenotype-genotype association papers because it is easier to standardize, it is widely available, at least spirometric values like FEV1, and it is easy to pool between multiple centers from different countries this data and do large population comparisons. So this is another finding from a very large study from the international PCD cohort that includes both retrospective and prospective data on lung function, where we see that patients with microtubular defects had a much worse lung function using FEV1Z scores with the GLI reference values in comparison with other groups, which were either the normal non-diagnostic ultrastructure or the dynein and arm defects. Again, even in this kind of very big studies, the possibility to have large groups to compare is because we are grouping many different genes under the functional phenotype group. This is the first longitudinal study in adults a few years ago that didn't just describe spirometry at baseline, at diagnosis, but followed it for several years and tried to see if the slope of the FEV1 decline different between the different ultrastructural defect groups. And this study from UK showed that, again, patients with microtubular defects not only had a lower baseline FEV1, but in that case presented as FEV1% predicted, it was also the annual decline for these patients was much steeper in comparison with the normal or inconclusive ultrastructural defects or with the DNA and arm defects. And several studies follow to actually prove that these findings were not random findings, but have been seen in different populations. So this is a large study from Italy, prospective and longitudinal, that also really followed this population for a period of time and studied that growth and lung function in comparison between groups. They presented the results separately in children and adults, which I find always very useful because we know that several adults with PCD have been diagnosed much late, sometimes even in adulthood, and their clinical characteristics differ a lot from the children. So they found that the children that they had the lowest BMI were children with central apparatus defects, and also they were the ones together with the microtubular disorganization that had the worst lung function. And the same finding also was for adults in that case. And their population, the biggest part of their population was genotype. So they also describe in the paper that the worst evolution of lung function from all the mutated genes was patients with CCDC39 or 40 mutations, and the best evolution that they had in their population with DNA 811. And I'm getting back to that North American cohort. I presented earlier their baseline cross-sectional data, and now we have their longitudinal data where they also really try to study the slopes. They used 4 different categories that you can see here in the graph: patients with outer DNA and arm defect, patients with outer and inner DNA and arm defect with the green, and then patients with central apparatus, microtubule disorganization, and inner DNA defects, and then normal ultrastructure. And what they found is, again, that the red graph, you can see the overall slope, the mean slope, which is the, can I open the pointer, which is this here, that it is much steeper for this group in comparison with the other three groups. And they had similar findings for the weight and the height. And specifically, their patients, the biggest part of their patients was also genotyped, and they found that this corresponded mostly to patients with a CCDC 39 and 40 mutation, and the best lung function and weight and height was in patients with DNase 5 defects. As I said, FEV1, it's easy, it's available everywhere, but we have recently started having more and more studies that assessed other outcomes. The Italian prospective cohort that I mentioned earlier also studied lung volumes in this publication for a couple of years ago. And this was the first study that did that, especially with longitudinal measurements. And they also showed, you can see here the three largest gene categories, gene groups that they had in the population, that the residual volume Z scores in the patients with microtubular disorganization and central apparatus defect had a much steeper increase, elevation rate, and the same also for the ratio of RV to total lung capacity. Compared to the other two groups. And that showed that these patients really had a greater hyperinflation with time. I have a few more slides. I will try to be on time. This is just some data presenting imaging comparisons between different groups from the American cohort. Again, it's a very new paper from a couple of months ago, and they really show that patients, again, from the same microtubular disorganization defect group had more mucus plugging and more percent of radiographic disease using CT score that was adapted from cystic fibrosis in comparison with the rest. I want to say a few more things on this study from Sumark et al. That included a really large population of children from UK, France, and the Netherlands. It used cross-sectional and retrospective data, but was characteristic is that instead of just comparing one specific outcome like lung function or the prevalence of one symptom, they really applied a data-driven hypothesis to try and describe phenotypes. And you can see here that they said that patients with central complex and DNA9 regulatory complex mutations, they had much worse lung function, FEV1 score, in comparison with the other patients. And they also found that patients with DNase 11, they had absence of neonatal respiratory distress in comparison with the other groups. They described also some findings related to clinical symptoms such as history of rhinitis and glue ear. However, one of the weaknesses of the study is that, of course, the clinical information were all assessed at diagnosis, and it didn't have the possibility to incorporate longitudinal data. And overall, what they found, which is, I think it is very new in comparison with the other data, although it doesn't provide us with different information, it has a very different approach, is that CCDC 39 and DNase 11 had the most distinct respiratory phenotype, while DNase5 was the most diverse group, which did not correspond in a specific phenotype. This is just a quick example to show that not all studies really replicate this kind of results. So this is from our own cohort that focuses on the upper airways, the EPIC-PCD study, and studied sinonasal and otologic disease in a large number of children and adults with PCD from 10 countries. And we did not find any differences using a combination of symptoms and clinical findings between the ultrastructural defect groups. I will not say, I'm a bit over with my time, so I will not say too much on that. I will just say that there has been studies published that specific mutations on these genes listed here, they have also been associated with normal nasal NO values, and this highlights the fact that we should not disregard patients with normal nasal NO as non-PCD. And about fertility, the things are a bit more complicated than the respiratory phenotype. We know that both for the sperm flagella, but also for the oviducts, the motile cilia play a big role, and the mutations lead to fertility problems. In the past, it was believed that almost all males with PCD have fertility issues, but it has been shown that specific proteins coded, but some genes like the DNase5 or the DNase11 have led to patients without fertility problems, while other mutations in genes like CCDC39 or 40 led to patients with infertility. That doesn't mean that all the patients having this mutation have or don't have fertility issues. As a summary, and to finish with my presentation, I just want to say that these studies that I presented, and in general studies for genotype-phenotype associations, they have, even the large ones, they have small sample sizes for most genes. We can have large groups for the most common genes, but for most of them, even grouping together, the numbers become very small, especially when we do comparisons by age group. And using this ultrastructural defect grouping, it helped so far a lot, but still might lead to oversimplification of some of these associations. And one of the main problems that we have to study genotype-phenotype associations is that the clinical data, especially data related to symptoms and not to outcomes as lung function, are not standardized and often are very incomplete from charts. So we do need very large and prospective studies to collect this information. Another issue that I want to raise is that do we really interpret sometimes in the newer study what we expect and want to see? So we know that patients with CCDC39 or 40, they have a worse respiratory phenotype, and that has been shown in several studies. But not all studies always that compare disease severity and different aspects have the same findings. And sometimes we have to not only focus on the evidence of association, but also keep in mind that there are studies that they do not find evidence, and try to think the reasons that this is the case. So what and this is my last slide, what it's important to know is that yes, genotypes, phenotypes, associations, they are important for researchers and clinicians to understand how PCD is expressed, and also to help to expect prognosis for a patient. There are several studies already, lots of published evidence for genotype-phenotype associations, but while few things are clear, most of them are not so straightforward. So as I said, we really need large international studies like registries or large cohorts. And ideally, we need an approach like this, the approach from UK, France, and the Netherlands that I showed, that use some clustering algorithms to describe phenotypes as a whole and not just compare separate characteristics. And as a last thing, we need to remember that patients with PCD, they are not just defined by their genotype. There are several other important factors that play a major role for the expression of the phenotype. And this can be environmental exposure, socioeconomic factors, other accompanying diseases, very importantly, differences in the management and health-related behaviors that they have, like exercise, for example. So this is where I will stop. I think I went slightly above my time, but hopefully not too much. And I stop sharing.
Thank you. I'll just carry on. So thank you so much, Myrona, for the beautiful clinical introduction to genopheno. That's very helpful. Can people see those slides? Yeah. So my job here is to try and keep to time as well. I'll aim for be stopping at the hour. So Katie, if you do remind me if I'm going over time, I have my eye on the clock. So I'm going to speak a little bit more about the functional biology underlying PCD and the genotype-phenotype correlations that we know about. And so to get straight onto that, we are all, I expect in this audience, familiar with the 9+2 structure of the motile cilia in our airways and these typical structures. And Myrona has shown this already and explained a lot about them. And these are the main features. So that's what I'll be talking about. But I'm going to go a little bit more into the more rare forms and some additional structures that we found. But very typically in PCD, or the most typical thing that we see is the loss of the outer or the outer and inner dining arms in patients. So with that typical 9+2 structure of the arms, the outer and inner dining arms coming off the peripheral doublets, the central pair doublet with the radial spokes and the necks and links in between those peripheral doublets. So as recently defined, class 1 defects are these ones here that affect these major structures in the airway and cause the most common defects, the ones that we're most aware of. So class 1 defects include loss of the outer arms, loss of the inner and outer arms, and microtubular disorganization and IDA defect, some cases of that. So I can explain a little bit more about the basis of these defects. So these are the classic, the slightly more classic forms. And in this slide, it's a little bit old slide. This doesn't contain all the genes that we know about now. This is a slide created by Marie Legendre from a teaching session that we did some years ago, just to explain these class 2 TEM defects. So we have things like the CCGC 39 and 40, this disarray of the microtubules and loss of the reduced inner arms can sometimes be called a class 2. With some of the genes that affect the central pair, there's a 9+0 cilium with the airway cilia completely lacking there. Central pair, or you can have 8+1 where one of the peripheral doublets shifts into the center. So these are the more complicated or slightly more rare mutations and defects that we can see, but there's also quite a lot of more subtle and normal EM results. So these are patients where we don't have structural defects, for example, DNH11 in some cases, DRC1, these necks and link genes. And we have ciliogenesis defects as well. So their structural classification for class 1 is ODA defect, ODA IDA, and the microtubular disorganization with IDAs missing. Class 2, that these aren't the so-called hallmarks, but they indicate a diagnosis if there's other supporting evidence: central pair complex defect, mislocalization of basal bodies with few or no cilia, the multiciliogenesis type of defect, microtubular disorganization with some IDAs present, and a sort of lesser absence of the outer dining arms affecting less of the cross sections, and combined ida-oda if there's less of the cross sections affected. So we get this slightly more subtle spectrum of defects that can indicate a diagnosis of PCD in common with other supporting evidence, and other supporting evidence can include genetics. So on top of the slicing through a cilium and looking at the cross-section, the arrival of EM tomography and 3D imaging and even atomic models that have started in ciliate organisms, and now we can see them emerging even for mammalian and for human cilia. These have dramatically changed what we know about the structure of cilia. And I just show this tetrahymena. This is a multiciliate organism. Just to show to sort of appreciate the power of cryo and tomography. So this is the peripheral doublets next to each other within the cilium, and you can actually see the outer dining arm stretching between the two. And in Chlamydomonas, you start to get a real high-level molecular definition of the outer dining arms and the inner dining arms and the radial spokes within this 96 nanometer repeat. And also the finer structure of the central pair microtubules. With this type of imaging has really resolved lots of new proteins and things that we weren't expecting. So just on the 96 nanometer repeat, all the way along the microtubules, we get the repetition of the outer dining arms, and there's 4 outer dining arms in this 96 nanometer repeat, and 1 outer dining arm per 24 nanometer repeat. And these are utterly conserved and vitally important for motility and cilia. So if that goes wrong, that's when you get a problem, and that's when you get PCD. So 2023 was this remarkable breakthrough of actually visualizing the first human axonemes with atomic models at this sort of resolution, moving from the ciliate organisms. And again, here's the 96 nanometer repeat, but this is what it looks like in human. There's just a bit of the radial spoke 3 that can't be, they don't have a model, so it's a slightly less organized picture, but the rest is a really high definition atomic model. And when you look at these structures like the radial spokes, they come in, there's 3 of them, and they're repeated within the 96 nanometer 1, 2, 3, 1, 2, 3, 1, 2, 3, all along the axoneme. We start to see PCD proteins. So mutations in these proteins are causing the disease that we know about. And there's actually these novel gene products that may well be new candidate genes. And one thing I want to just talk about are the ruler proteins. These have really come to the fore as causing severe PCD. So I want to talk a little bit more about the biology of that correlation. Why do they cause more severe disease? And then within the peripheral microtubules, so the 9 peripheral microtubules going around, we start to see, using these atomic models, actually structures sitting within the microtubules. So these inner microtubular proteins decorate the microtubules inside. Those really weren't appreciated until very recent years. And mutations in these components can also be forms of PCD, can also cause forms of PCD. So the MIPs that I just mentioned, they're generally associated with more mild forms of PCD or slightly more, what can I say, slightly more unusual forms that we can't actually call PCD, and we have to call them laterality defect with respiratory symptoms or these sorts of things. And I've just listed here MNS1, CCDC 11, CCDC 19, WDR 16, and NCUR. So these are the 5 that we know about that have been associated to some degree with either PCD or airway symptoms, even if we can't define a really concrete EM defect and get all the precise diagnosis for what we want for PCD sorted out. So they're quite highly associated with laterality defects. In fact, CCDC 11 has been known for a long time as isolated situs inversus gene until people started to realise that you can get airway symptoms. So it appears that these MIT may have more of an effect on the nodal cilia. So nodal cilia are cilia in the embryonic node that set up our left-right axis. They are outside the lung and they lack the central pair. So they may be more vulnerable to loss of these inner, these MIP proteins than the airway cilia. In the node, you get fewer cilia, they lack the central pair, they may be less stable, they have a rotational beat. So there may be reasons why these are primarily or very often seen as patients with situs inversus and laterality defects, and with the milder airway symptoms becoming more associated. So they're quite difficult to classify as classic PCD. However, the ruler proteins are now reported by multiple groups to cause severe disease, and in all studies they really stick out as having dramatically severe respiratory symptoms. And the reason is that the loss of these proteins causes a complete loss of that 96 nanometer repeat, and this has really only been properly pinned down this year with the visualization of PCD patients with CCD 3940 mutations. So you can get an idea of their importance in yellow. So CCDC 3940, they're coiled heterodimers. They bind each other, they're binding partners for each other. So if one is absent, then the other is not going to work. And their role, they stretch out the whole way along these peripheral microtubules. They have a vital role to set up this 96 nanometer repeat. So the outer dining arms being placed in their pores all the way along and creating the forward and backward stereo beating that's completely reliant on these two proteins. So just an example, and I think Mathieu is on the call. So this is Mathieu Bottier's beautiful algorithm for measuring, very sensitive measurement of cilia motility. So, and this is the atomic modelling by Alan Brown in Harvard. So we have, as an example, patients, a comparison between patients with ODAD1 mutations, and ODAD1 is a component of the outer dining arm docking complex. So if you look at your peripheral doublets, all 9 of them, you'll see the little docking complex, and that's where the outer dining arm attaches. So we were able to, I mean, it's quite difficult to freeze patient cilia in enough quantities to be able to send them to Alan Brown in Boston to create an atomic model. So this is really fantastic work. So comparing a patient with an outer dining arm defect on the top there with CCDC 39 and CCDC 40 patients, and where CCDC 39 and 40 sit is right here all the way along the length. This is the cross section of the peripheral doublet there all the way along the length. So with ODAD1, you actually get static flickering cilia because you lost the outer dining arm, and you can see the loss of the 24 nanometer density in these patients. This is a wild type. So looking along, you can see that they do have their CCD 3940, but they completely lack the 24 nanometer density, and that's over here. That's the outer dining arm. So they lack outer dining arms, and thus they are static. In CCD 3940, you get actually a little bit of motility, and you might think, why isn't that giving a better mucus clearance than a completely static cilia? But they are really truly messed up. So the whole of the outer dining arm, inner dining arm, the whole thing, the 96 nanometer density is lost for positioning of that, and that has a devastating effect on motility. So just in our familiar transmission EM microscopy, we can see that the ODAD1 lacks the outer dining arms. CCDC40 has a bit of a mixture, missing outers, inners, eccentric, central pairs, microtubular transposition. It's like the whole of the structure is completely messed up, and that's clearly much more detrimental to airway cilia. So To summarize a lot of information, this is a wonderful picture, again created by Marie Legendre in a review written in 2021. And what I've done here is just updated it with the new genes. So green genes, and I'm not claiming this is even the complete list of PCD genes, but we have several central pair complex genes, those MIPs, an outer dining arm component, inner dining arm component, and a couple of docking complex components, and also genes involved in actual generation of the multicilia. So we have several new genes literally just in the space of a couple of years where we're starting to link these to PCD, and that's very much through the power of next generation sequencing and people really getting traction with looking at large cohorts with DNA and now RNA sequencing to determine the mutations. So I just want to say that we now have a really complicated story of 60 and counting PCD genes. Some of them we are more familiar with and some of them are not. So there's a whole bunch of factors that assemble the dining arm in the cilia. In the cell body of the multiciliated cells, and these are the DNAFs. We have these multiciliogenesis genes, and just I'll mention those a bit more in a minute, their function. But these are also they're called various things. These genes, they're also called the genes that cause RGMC, which is reduced generation of multiple motile cilia. So patients with mutations in these genes lack a cilia. They have sparse cilia numbers and shortened cilia with these various genes. And then we have the structural components, including genes that are assembling inner dining arms and central pair components and these finer structures, including the nexin links, the radial spokes, and the head and stalk, and the CCDC39 that sits there but goes all the way along the axoneme. So we still got 15 to 20% of patients that aren't solved and have normal or difficult EM, where we want to know if they're PCD or not. And this is much more solved by genetics. I'm just showing a couple of examples here. So hydin is a central pair component and quite a common cause of PCD, and probably more common than we realise, probably quite underdiagnosed. Because by conventional EM, you don't see that much difference in patients. They can look really quite normal. And that's because the central pair just lacks this tiny C2b projection that you only really appreciate if you do electron tomography. The nexin link, these are the links between the peripheral doublets. So normally you see a nice nexin link, and that's a multi-protein complex with lots of proteins that can cause PCD if mutated, and a couple of them are called DRC1 and DRC2. So by 3D, you can see the nexin link is completely absent. So these patients have a sort of really normal looking cilium, but it's not normal at all, and it doesn't beat properly. And DNH11, why is DNH11 causing apparently a very undisturbed 9+2 structure when we know that it's a very big protein component of the outer dining arm? It's because it's sort of tucked away. It's in a bit of a knuckle. So these are control 3D images, and DNH11 in a control is when you're missing DNH11, I haven't shown the control yet, what you get is a kind of hole in the arm, but when you look up sideways view, it just looks quite normal because it's sort of hidden by the presence still there of DNH5. So this sort of structure doesn't look like that much different from that structure, and you have to do tomography to find that out. DNH9 is also quite it's a more rare form of PCD mutations. It's similarly a large outer dining arm protein, but what became apparent was that it only affects cilia in the distal, the outer dining arm structures in the distal cilium. So DNH9 is only in type 2 outer dining arms, and they're at the top of the cilium. Type 1 dining arms down at the bottom of the cilium have DNH11 instead of DNH9. So in a normal healthy cilium, you get DNH9 outer dining arms attached to the microtubule, and here you get DNH11 containing outer dining arms. In a patient with a DNH9 mutation, you still have half your component of dining arms. You've got the type 1 with the DNH11 still there and unaffected, but up at the top, your DNH9 containing outer arms are absent, and that causes a general reduction of the whole arm. So it's quite nice to look at this, and this is work done from the home at Omron lab, where they visualize this quite nicely. That in a healthy cilium, you have this forward and backward stroke. DNH5 pretty much static. DNH11 and DNH9 retain quite a lot of movement, but it's not functional movement, and it causes disease. So here's the healthy control cilia, and I hope you can see that in DNH9, where they only have the top cilia, outer dining arms affected, the bottom of the cilia is moving and the top is just being dragged along. In contrast, in DNH11 patients, the bottom is static because they've lost DNH11 protein, but the DNH9 outer dining arms can still move. So it's really quite subtle and really quite hard to diagnose. And it may be why they are underdiagnosed, but it may also explain why these patients are relatively mild and affected. So the concept is, and Johanna Wright has written this nice, got a nice cartoon that these various different motilities caused by very specific gene mutations and structural defects, from wild type to stiff and static, discoordinated, completely immotile and rotatory. So when you lose your central pair, when you lose your outer dining and inner dining arms, when you lose your outer dining arms, and when you have your the CCDC 39 or 40 type variants. So briefly, outer dining arm and outer dining arm docking complex, you get this statics flickering. Central pair radial spoke, necks and links, inner arm, these are involved in signal transduction. So rather than actually powering the beat as the outer dining arms do, these do more to regulate the waveform and generate the ciliary beat pattern. So for example, central pair hide-in, you get an uncoordinated and rotatory movement. The dining assembly factors lost where the outer and inner dining arm is lost. So dining assembly factors, for unknown reasons, you lose both the inner and outer, and you're very static. CCDC 39, very severe disease and very uncoordinated motility. These are reduced generation of motile cilia or multiciliogenesis or oligocilia defects. A very severe disease, shortened and reduced cilia numbers caused by these various genes, CCNO, multicillin, FOXJ1, NEP10, TP73, IFT74 is a new one. And then we have a few forms where there are syndromic features, and I'll just have a follow-up slide just about those. So that's quite a lot of detail. Amelia Schumacher and colleagues have written a nice review recently, already out of date, of course, because of the rate of gene discovery. But I just want to show this to say that structure and motility defects then link, of course, to clinical phenotype, genotype correlations. So, for example, if you have your Nexon link mutation sitting in between here, and I showed you DRC1 and DRC2, you can get some microtubular disorganization, but not very much. It's quite subtle, and it's quite a subtle effect on the ciliary beat. And there is no laterality defect. The Nexon link has no effect on the nodal cilia during embryogenesis. It has an effect on airway cilia. But you know that with Nexon link mutations, patients are not likely to get laterality defects and therefore not likely to get congenital heart defects and heterotaxy. So there's a lot of similar correlations here, and I won't go into them in detail because we had a nice summary of quite a lot of that from Myrona. So syndromic PCD and RGMC can affect multicilia, and also some are emerging where primary cilia So the primary cilia that do a lot of signalling and are very ubiquitous in the body and cause primary ciliopathies. We now have a couple of primary ciliopathy related genes that are emerging that cause PCD. So because these are so rare, again, I won't go into them, but we have these RGMC forms that I've mentioned, rare syndromic RPGR, PCD with retinal dystrophy, OFD1, oral facial digital syndrome plus PCD, TP73 is RGMC with lissencephaly, so malformation in the brain, IFT74 RGMC plus a skeletal ciliopathy called Jones syndrome. And just as an example, IFT74 is an important trafficking protein in cilia. So that's why it causes defects in both primary and motile cilia. And those are the multi-ciliogenesis regulators, and I refer you to Legendre review again, 2021, to look at a little bit more into those if you're interested in the RGMC generation. Okay, so I'm already nearly at the hour. I'm going to take perhaps 5 more minutes with some thoughts. Myrona mentioned that a whole bunch of genes are not related to laterality. They don't cause laterality because they don't affect the node cilia. So these are really nicely clear. And my point in showing this study from Best et al. Is just to reiterate that large studies are required. So there's so many multiple genes and multiple mutations in PCD. We really need large studies, and this probably isn't large anymore at the time in 2019. 370 patients with their situs having actually been determined, it's quite large. And then it's really clear cut. You can see exactly which genes are not associated with abnormal situs. As well as gene groups, there's of course allelic variation. So within a particular gene, for example, DNH5 shown here, just a simple example from a little while ago, one of the original papers on DNH5. Just allelic variation means within that gene, different gene mutations can have different effects. So in a healthy control, here's green, the marker of the cilia, in the airway cilia when you put them on a slide, and DNH5 is an autodyne component that goes all the way up and down the axoneme, so very nice co-localization. In a patient that carrying a DNH5 of actually at the time would have been a variant of uncertain significance because it's just an amino acid swap, an arginine to a leucine, plus a null allele frame shift. What you see is no DNH5 in the cilia anymore. Having something in the cell body, which is here, that's not unusual. But the main point here is that the protein is completely not able to assemble in the cilia. So we know that missense mutation is actually effectively a null allele that's doing the same sort of job. Whereas here we have 2 splicing mutations in a patient, and actually quite a lot of DNH5 is still retained in the cilia. And there would have been a slightly less of an effect on the motility in this patient. So this patient quite potentially has milder effects. I think one patient on its own wouldn't be able to say, but just my point here is that different mutations can cause quite dramatically different effects where protein absent and protein present to some extent. How we measure variance, we have to be quite careful. This is an EM survey that was published from our lab in collaboration with the Royal Brompton. So these are all genes, gene mutations in outer dyning arm defect, in outer dyning arm genes, sorry, and these are dyning arm assembly genes. And I mentioned that with ODA gene mutations, you just expect ODA loss, which is marked here in blue. With dyning assembly, you should expect both IDA and ODA, so in red, both the arms missing. But I just want to say that I don't know what date the EM was done, but for example, in a dyning assembly factor gene DNF3, here we have completely, all cross sections completely lack both arms, but here we have a good lot that was sort of registered to only lack the outer dyning arm. So you could easily mix that up with you could get that confused with an outer dyning arm gene defect. So the genetics here is quite good. And similarly here we have a DNH5 variant in a patient whereby, and I notice it's probably a VUS and amino acid switch, where there's actually both arms present in a lot of the cross section. So again, the genetics here is helping a lot with that difficult diagnosis. So my point here is that you have to be quite careful with your clinical measures, and you know, we're getting better at all these clinical measures as we go along. So for example, in EM, we would now be paying attention to whether we're looking at the top or the bottom of the cilia. CCDC103, this is another dynein assembly factor where outer and inner dynein arm loss is happening when you get a null allele, but the disease is really dominated by a single mutation, a founder effect mutation, a histine to proline change at amino acid 154. And this is certainly a hypermorphic mutation. So when you have a healthy control, you can see the cilia beating, and in a patient, there's a fair amount of retained motility. When with a gene like this, where both inner and outer dynein arms are lost, you'd really expect to have very static cilia. So this is particularly associated with this amino acid change, which is very enriched globally, and we have a lot of South Asian origin families based in the UK with this mutation. And what you see, this was written up by Schumacher et al. In 2018, they have some normal measures. So size inversus, no. Neonatal distress, yes. Rhinitis, but no loss of outer dynein arms, and the NO level quite high. So patients with this particular mutation who are homozygous, you can have two siblings and one sibling with the exact same homozygous mutations being discharged from clinic. So that's sort of poor recognition of this rather intermediate phenotype. But it certainly does cause disease. So in South Asian origin populations, we have this set of patients with that particular mutation. Here's a comparator group of the same background, British Asians, but PCD caused by different genes, and they really have a similar drop in their lung function. But in the CCDC103s, we can see a really high level of size inversus, 81% versus 56 in the comparator PCD group. Again, higher nitric oxide on average, quite high, and ciliary beating quite a lot. So this tends to suggest that maybe you only get picked up, or you mostly get picked up, if you happen to have laterality problems. That's not 100% in these patients, and perhaps there's quite an underdiagnosis of this particular mutation. So I just have a couple of finishing off slides that really summarize quite a lot of geno-pheno at the biological level. So do forgive me, I can't really go through them in great detail, but several genes cause a problem with the central pair, radial spokes, and some of these radial spoke and hide in as well would be on here. So if you have a central pair defect, you're not going to affect your nodal embryonic cilia. You don't expect to have size inversus or part defects. With RPGR syndromic, that's to do with photoreceptor maintenance. So you also won't get size inversus and heart defects. You may get rare PCD. These are Nexin link. Nexin link again is not going to affect situs. There are a couple of genes that are associated with outer dynein arm defects, but that there seems to be some redundancy in sperm because they escape or have no male infertility. And photoreceptor RPGR patients also no infertility noted, but that's low numbers. Multiciliogenesis patients, multiciliogenesis is not relevant for male sperm, so they're not going to have male infertility. They will have worse lung function, more hydrocephalus, but they will not have size abnormalities because again they are multicilia genes, and the nodal cilia that determine our laterality are not multicilia, they're monocilia. And CCDC39 famously the worst lung function. I feel like I can't go through all these. These are some examples of milder and subtle forms. And I do refer you to a really great review from Brennan et al. That's come out not so long ago, 2021, very nice geno-pheno correlations. There are some more recent single cases, so 2022, Ostrowski group, ODAV1, that's a docking complex milder phenotype. Some of these are really not understood, GAS2 2 milder PCD, but we do have emerging forms of subtle PCD, and I've managed to talk about just a few of them. So we have made great progress with our 60 gene products. We have these fantastic collaborations going on, including BEAT-PCD and lung. And this is a lovely poster from Johanna, and hopefully coming out in publication soon, where we have this remarkable ability from the European consortiums to actually start to look at specific mutations and where they are enriched across Europe. So I told you about an ODAD1 mutation. It's used to be called CCDC114, where there's no male infertility. We know that there's a great hotspot of those in Holland, so we can say that those males will be able to have children with PCD, and that's across the world. And then there's common mutations, and so this is going to be more and more interesting to be able to make these correlations. So in conclusion, we have multiple genotype correlations and phenotypes. But the truth is, this variability is really largely not understood at all. And we have so many multiple variants of uncertain significance and complications in PCD and difficulties with the genotyping and phenotyping. So this is a huge area we're really just starting on. But we have some very good suggestions now, and we certainly know some of the more and less severe forms. So structure and function of cilia clearly linked an impact upon mucus clearance and PCD severity. Our understanding is going to grow. We have these links, as I mentioned, CDC39 and 40 severe, DNH11, DH9 more mild. And important to remember, yes, gene type, gene group, but also mutation type within each gene is going to be vitally important. And that's where we really have a big job to understand mutations and particularly these variants of uncertain significance. We have to be careful with our older data, and we have to get better with sort of recording and documenting. So, you know, for example, I've just used this simple thing about we need to look at the top and bottom of cilia when we're trying to make an EM diagnosis. Genetics is definitely not a gold standard yet, but with 60 genes, we're getting better towards 80 and maybe 90 and maybe one day a gold standard, but we still are reliant on multiple tests. And in particular, if you tend to have an unresolved genetics, you very often have an unresolved phenotype. We have many cases where you find one allele, one mutation, and you don't find the second one. So cilia functional screening is very important. And I think I'm going to stop there. Thank you very much. Stop sharing.
Thank you very much, Hannah.
I'm sorry, I went a little bit over, so apologies. Yeah.
No problem at all. We still have time for our Q&A. So excellent talk by both of you. Thank you very much. We start the Q&A with questions from the audience. Christopher says, thank you, and this question goes to Hannah. Both excellent talks. I understand that the 100,000 Genomes projects highlighted the 4% of patients with bronchiectasis, but no diagnosis or other suspicion of PCD did in fact have PCD-associated gene mutations. Did they look at the full 60 genes? Which genes did these patients have?
Yes, great question. So in the 100,000 Genomes project, the diagnosis of PCD was superb, really high, but not all of them. I think there's still a couple. In bronchiectasis, very interesting that maybe 10% are hidden PCD patients. And did they look at the 60 genes? That is a fantastic question. They absolutely did not. So a genome project, the real truth behind a genome project is it's all based on gene panels. And more to the point, it's based on clinical gene panels. So they have a traffic light system, green, amber, red. So some of those genes that I have talked about, you know, MNS1, these ones where there's one family, we don't know if it's really PCD, etc. They would definitely be red and amber genes, and they are not looked at by 100,000. So there's definitely underdiagnosis, absolutely. And there's not a lot of money. There's these so-called GeCIP groups, which are people like me who are supposed to follow up and do all the functional work. And, you know, I wish they'd give us the money to do that. But no, completely right. They've looked at a fraction of genes and the overall 100,000 is quite a low, maybe 20% diagnosis across the board. So lots of room to do more.
Thank you, Hannah. So now we have a question from Lee Baker. Thank you both for the informative presentations. I have a question regarding the classification of PCD. Would something like the inhibition or alteration of the MAPK pathways that lead to abnormal cilia length count as a form of PCD?
I am not involved in any of these guidelines and groups, so I'm just a humble. I wouldn't even presume to say. I think that's very difficult. So we talk about RGMC. Some people call it oligocilia. Some people call it cilioplasia. Some people say, why don't you just call it PCD? You don't get mucus flow. So very complicated. So as part of the ClinGen consortium, we are trying to find every single gene that has any of these associations and take a formal categorization to see what's the evidence. Does this look like PCD? Have model organisms been looked at? You know, have you looked at the EM? So we are trying to do a more formal and rigorous classification. But yeah, you know, TP73 and some of these ones that are, even in the title of the paper, they're not calling them PCD. So I feel like maybe others who are not me might decide these things. I'd love to know what you think, Myrona. You join these groups more than me.
And not the specific one, but I think it's really not clear cut. I think the work that you're doing now with ClinGen will help a lot to really discuss and decide if we can call that PCD or not, because there are these different opinions if really we should just use a big umbrella for all the ciliopathies, or if we should really expect at least a mostly typical phenotype, at least respiratory phenotype, to call something PCD. So as far as I know, this is not at the moment characterized PCD in most publications, but hopefully with the work that you're doing with ClinGen, we can really have a better guide of what we can call PCD and what not.
So ClinGen, just to stress, has got clinical experts and molecular experts and these biocurators, but it's still very challenging. Yeah.
Thank you very much. Now I have a question for Myrona. The studies that you presented with phenotype and genotype associations, the phenotypes mainly focused on lung function or growth. So do you think next genotype studies should repeat this in larger cohorts, or do you think they should focus on other phenotypes?
I think it's normal that most of these studies really focused on growth and lung function, and as I mentioned, easier to measure clinical outcomes. In my opinion, we need to follow phenotype approaches that have been followed in other diseases. So phenotyping is something more complex in PCD, but it's not something unique. We have seen it in common and rare diseases. We have seen it in diseases like asthma too, and it's not necessarily, as I said, depending on the genotype only. We expect that it will be more dependent on PCD, but I think we need to follow this kind of approaches like the clustering algorithms, where we can incorporate functional information like the ultrastructural defect, information on clinical measurements like lung function and growth, but also some information on disease severity from the symptoms. And it might be also the case that we need to accept at some point that some very common symptoms like rhinitis, for example, the study that I presented from Amelia from UK and France and Netherlands, what had struck me as very impressive in that study was that they found that there was a subgroup of patients with very low prevalence of history of rhinitis. However, we see in very large data set that more than 90% of the patients report rhinitis quite severe at some point of their life. So a point is, at what time point in the life would you get these patients? When you have cross-sectional data, this is a bit difficult to pinpoint. And another point is also that maybe characteristics that they're very common or very uncommon are not great to group phenotypes, because if almost all the patients that we have in a population has a characteristic, it will not help us to differentiate. So I think we need to adopt more and more this kind of approaches with clustering of distinct groups and select different kind of characteristics that will help us to differentiate.
Yeah. Can I just add, I completely agree with you. I was a little bit involved in that topological analysis. And in fact, that's thanks to Jane, Lucas and Katie's group that we had access to that amazing software, and we've lost it actually. So we need to pay money to get it back. But it was completely model free and remarkably powerful. But even with 400 genotyped, you know, we threw out a lot of patients with an incomplete genotype. We were really strict. 400 genotype patients. We still couldn't get genotype, a sort of very clear genotype phenotype, and we couldn't get mutational, but you know, mutation type specific. Do null alleles cause a worse disease than Mitchison? So I don't know what kind of size. It will need a lot bigger. It would be really nice to think that the European consortia in America can get together just to make bigger and bigger groups. And then you need actually a confirmation group as well. So you need to double your size. Yeah, but it was really powerful. Yeah.
Thank you both. So I have a question now, and it kind of follows on from you talking about the need for large data sets. So for people who don't know, how can they contribute their patients to registries or to ongoing studies?
I think that one of the, Hannah already presented at the end of her presentation the data set from the ERN lung registry, which is at the moment the largest international registry, especially with European data, but also with some countries outside of Europe that contribute. I think contacting the group in Münster that has hosted this database where we all contribute data would be the best way to contribute. And then depending on the question, there are also other research-based projects, not registries, like cohort studies. For example, I shortly mentioned the upper airways cohort that we ran with Yinting, that they are not designed for genotype association, but they can focus on some areas where, example, the registry does not focus. So depending on what interest the people have, I think that they should contact for sure the registry. There is a question also in the chat about the contact detail for patient data. I think that you, Kate and Yinting, have the email from Münster, like Petra's email, I think would be best, or Johanna's. You can share with the people and they can just contact them and get all the information needed for that. This is the only way that we can really go forward for this kind of studies. Otherwise, as Hannah mentioned, even with large data sets, you will never be able to have detailed enough numbers to really see, especially if we go into specific mutations, as Hannah, you mentioned. We will never really have these numbers, even with collaboration of 3 or 4 or 5 centers. We really need worldwide studies.
Thank you very much to you both. And yes, the follow-up question was actually what you just mentioned, Myrona, that Christopher asked if we can share the contact details to contribute patient data. Yes, certainly, and I think
I will put it in the chat.
Perfect. Yes, that'll be the most direct way. Excellent. All right. So I think thank you for putting that in the chat, Myrona. Since there aren't any more questions, we should close the session then. So thank you both very much again for this excellent talk. There were a time, I think 37 people who joined, and I think that's an excellent number for a live session, and even more people usually watch the recordings anyway. And
thank you for inviting us.
Yeah, thank you. That was great.
No pleasure. Thank you very much. And for those who have not clicked on the Padlet yet, we'd like to hear your feedback on this session, and we also collect your ideas and opinions for future sessions as well. And for the next expert talk, you can look forward to fertility in PCD, which will happen December. We will tell you the exact date and when we have fixed this with the speakers, but it'll be probably around mid-December. So thank you all very much for joining.